Good afternoon and welcome to today's special event, the November 2025 edition of Science Sundays. I'm John Beacom. I'm the chair of the Science Sundays Committee, representing departments from all around the College of Arts and Sciences. Science Sundays is one of the premier outreach events of the College of Arts and Sciences, and you might ask, why do we do it? The fundamental point is that knowledge unshared is like knowledge undiscovered. And not only do we want to share the things we find out with our colleagues in professional journals and other ways like that, and not only do we want to share what we find out with our students through our classrooms and research advising, but we also want to share it with the public. And that's why we're here today, and I'm delighted to see all of you here for that as well. Today's an extra special day because we're collaborating with the Center for RNA Biology, like we do every November, on a joint public lecture, and because we have a rare return speaker from Science Sundays from 10 years ago. And I will let my colleague Corinne explain all about that.
All right, thank you. Well, I also want to welcome you to this very special event. And again, my name is Corinne Musier-Forsyth, and I'm the current director of the Center for RNA Biology. This event is named in honor of Dan Schoenberg, who was our founding director, and Dan is here in the audience, and he's going to be saying a few words at the end. So the Center for RNA Biology consists of almost 50 labs that are here at OSU's main campus and also at Nationwide Children's Hospital. And we work in all areas of RNA biology, from sort of fundamental small RNA discovery to RNA structure, RNA viruses, and RNA and disease and therapeutics. And so RNA is really, one of the central players in a lot of the most exciting research discoveries over the past decades. And today we have I think a rare honor to hear from one of the pioneers in RNA biology, and really, I would say, one of the fathers of modern RNA biology. So I think you're really going to enjoy this lecture. But we want you to stay and hang out after the lecture. Just take a left out of the auditorium here, and you'll see refreshments, delicious food and coffee, and also, most importantly, posters. So we have our trainees, undergraduates, graduate students, and postdocs presenting their work on RNA biology that they're doing right here at Ohio State. And so they're really excited to tell you about their research, their discoveries, and their goals for the future. So I hope that you hang out and interact with our trainees. I think you'll really enjoy doing that. So now I want to introduce Dr. Wen Tang, who is a professor in the Department of Biological Chemistry and Pharmacology, and he's going to introduce today's distinguished speaker. So thank you.
Thanks, Corinne. Welcome, everyone. I see boys and girls here, so you are the future of science. It's really great to have you here. It's my distinct honor to introduce Dr. Craig Mello. Craig is a distinguished professor at UMass Chan Medical School and a member of National Academy of Science. His research explores how genetic information is controlled. That is, how some genes are turned on and some genes are turned off at the right time and in the right place. Using these tiny worms called cyanogans, his lab discovered a process called RNA interference. RNA interference acts as a switch to, turn off specific genes. The genes can be, genes from viruses or genes related to human diseases. As you can imagine, his discovery opens doors to developing treatment for human diseases. For his groundbreaking work, he and his collaborators received many prestigious awards, including the ultimate recognition, that is Nobel Prize in medicine and physiology. So beyond his extraordinary scientific achievements, Craig is a generous mentor and a supportive colleague. He has trained and inspired countless young scientists like me, and many of them are leading their own research programs in biotech companies and in academia, and two of them actually here in Ohio. So on a personal note, I find Craig's passion for science is truly infectious. He is full of interesting ideas and eager to share with them. I'm sure you will agree with me after hearing his talk. So with that, please join me welcome Dr. Craig Mello.
Well, hello, everyone. It's wonderful to be back. I absolutely love what you folks are doing here in organizing this opportunity for scientists to interact with non-scientists, especially the kids, because, well, in a way, the kids are the future, but also they kind of like naturally are curious, and curiosity is what science is all about. So this title is a little bit unusual. I wanted to capture something that is a realization that seems to be still sort of dawning on me and a lot of my colleagues who work in this field. Living things thrive on information. Information is like a fire that's burning. It's sort of the central fire burning in all living things. It's where the diversity of biological mechanisms springs from the information that's inside of every cell. And it's fascinating because the information moves. It doesn't stay put. And it's like an inferno with the information spreading and jumping from organism to organism. So I feel like RNA interference, which is one little mechanism for controlling information, is just a tiny spark in this incredible inferno of information that is the biological, the living state. And so I'm going to try to convey some of that excitement in my talk today. So for me personally, this journey in science, well, I knew I wanted to be a scientist, I just wasn't sure which kind. And I was reading the newspaper one day, and I saw this article when I was 16 and waiting for the school bus, eating my cereal or something, and I could not believe it. It was so cool. They had taken a human gene that encodes the protein insulin, and they had put that gene into a bacterial cell, and the bacteria could read the human code and make the human protein. So this is an incredibly useful tool. And the article was all about how now we could make human insulin, instead of having to take and extract insulin from pigs or cows, we could actually make human insulin in bacteria. But I read that whole article looking for where it would say, isn't it amazing that bacteria can read the human genetic code? Why is that? Billions, I was taught, my dad was a paleontologist at the Smithsonian, and I knew living things were really, really old. And I also knew that languages change rapidly. Why is the language the same after billions of years of evolution? To me, that sparked my interest in how organisms handle information. Why is it universal? And I felt like this was just something that I really wanted to explore. Now, here's the real Nobel Prize winner here, this tiny little animal. This animal is so simple, relatively. It has only 1,000 cells, and yet it shared 3 billion years of common ancestry with humans. It has neurons just like ours. It has intestinal cells just like ours. It has inner mechanisms for expressing genes that are just like ours. So why not work on this organism? And some of you do. Some of the labs here, like Wen's, are working on this beautiful animal. And I'll zoom in. I'll zoom in a minute. This is, I wanted to put I wanted to learn how organisms handle information. In order to do that, you have to be able to put the information back in. And so as my graduate project, when I was getting my PhD, I started working on this methodology that you can see right here. The needle is inserted into the gonad, directly where the germ cells are forming. And inside the needle, there's a DNA solution. And I'm delivering the DNA into the animal directly into what we call the germline. This is the lineage that makes the egg and the sperm. It's also the immortal lineage. It can go on and on and on. Living things on this planet are so ancient that the genes and the mechanisms inside of us are like living fossils. They are truly, truly ancient. On the time scale of the universe, life on this planet is older than many stars. Many stars only live about 1 billion years, the bigger ones. It's incredible. The living things are full of really interesting ancient mechanisms. So what better way to study how they handle information than to put it back into the animal? Well, it had never been done before. So it turned out to be really, really hard to get this to work. And that in a way, I always say, you know, your bad luck is actually good luck. You just don't know it yet. I was doing this, and so was Andrew Fire, separately. The two of us didn't know each other at the time, or we knew of each other, but thought of each other as competitors. So we're both trying to develop this methodology, and it turned out to be really hard. It took years. And over that time of having that difficulty, of trying things and having them fail, Andy and I finally got to start talking. You know, because when something doesn't work and you can't do it yourself, you start like, you know, opening up a little bit more to other people. Yeah, I tried that and it didn't work. How about you? What have you tried? We got to know each other. We got to start working together, and together we developed this methodology. Now, that also set the ground for our collaboration, which led to the Nobel Prize. Because when you start putting information into an organism, guess what? They've seen this stuff before. This is not new. It was first time humans had put information back into an animal, but animals are constantly exposed to information. Everyone in this room, I can say it pretty confidently, was infected by COVID. I got it 3 times. I don't know about you guys. Fortunately, I had a vaccine before I got it, so it was pretty mild. But that was 30,000 nucleotides of genetic code. That arose in one place in China, probably jumped from bats, and it spread to every person on the planet in the space of a year. Information flows horizontally. And organisms respond to it. So when we started putting information back into the animal didn't just take it. It actually reacted to that information, and it did stuff, cool stuff. Here's just a more higher resolution. You can see that the germline right here has multiple little nuclei that contain the DNA arrayed here around a common cytoplasm, and this is like a factory that's making the cytoplasm that goes into these eggs. So this animal is kind of politically controversial animal, I hate to tell you. It's a hermaphrodite. But even worse than that, it starts its life as a male, and then it transitions to becoming female. So first it makes sperm, and then it makes eggs. I'm hoping the Trump administration doesn't find out about that. But here this movie kind of shows sort of the dynamics of this incredible process. And I want to just point out a few of the features here of the ovary. Because, you know, this may seem like a worm, you know, is it relevant to humans? Humans and worms resided within an ovary, their ancestors did, where one of the eggs gave rise to nematodes like this guy, and another one of the eggs from that same gonad about 1 billion years ago gave rise to the human lineage. The genes that regulate oogenesis are conserved between humans and worms, a lot of them. Anyway, look at the green stuff. This is incredible. Do you know what that is? That's called mitochondria. And they are the remnants, and actually the living remnants, of a free-living organism that blended with an ancestral cell, probably more than two, we don't even know because there's no fossil record at this scale. So we're all guessing here, but these are informed guesses that are based on genetic sequences. So we can determine the genetic sequences of mitochondria and of organisms all over the world now because we can do this DNA sequencing. If you don't know what that is, it's just basically reading the letters in the genetic code. And so we can look at these and we can see that mitochondria are shared by plants and animals, and essentially are really, really old. Fungi have mitochondria, but all of them derive from the same symbiotic merger that happened. Organisms that were maybe infecting a cell became sort of a symbiont or adopted. It's called the endosymbiont theory. They were inside the cell and they gradually became blended. The organisms became merged. And what happened? The DNA, these cells, these green cells, used to have their whole genome. They used to be able to make everything they needed. The genome is what we call the DNA, the full copy of DNA that's necessary for an organism is called the genome. They used to have all the genes necessary for making a bacterial cell, but now they have only like 16 genes left because their DNA moved into the nuclear genome over probably millions and millions of years of evolution. Gene after gene after gene was transferred, and now the DNA that makes these cells function is actually inside the nuclear genome. In other words, the human genome and the worm genome is filled with bacterial DNA. The genes have been moved horizontally. In other words, again, this is living things have been in an information inferno for billions of years. They are really good at handling information. Information moves horizontally all the time. There is no such thing as a non-genetically modified organism. Everything is constantly being genetically modified. That's what viruses do, but not only that, our cells are really good at it too. They can capture information horizontally from other sources. And so that process played out over hundreds of millions of years probably to give us this relationship where we now have a blended organism. Again, the concept here is that living things handle information with incredibly sophisticated mechanisms that we are still just beginning to understand. And I want to, in relation to that, I want to draw your attention to these amazing red little blobs that are surrounding all the nuclei. Do you see those? Well, my mom's an artist, and she took one look at those back when I first started working on this animal, and she said, "Craig, what are those red things?" And I said, "Mom, nobody knows." And she said, "Well, you should work on those. Those are going to be important." And she was right. Because inside of each of those, guess what's sitting there? The key factors that mediate RNA interference reside in those little granules. In the germline, they are super upregulated. They are search engines, and they're sitting here in these little liquid-like droplets on the outside of each cell. And as information flows out of the nuclear genome in the form of messenger RNA. And I'll explain you're going to get a crash course in biology whether you like it or not. As the messages exit, every single message gets scanned by a search engine. It's searching, it's trying to determine if this is safe, is this my own information, is it good, is it okay, or should I silence it? That's all happening in those incredible little red granules. Living things are incredible. They are doing computations. They are doing things at incredible speeds to handle information. We don't appreciate this. We really don't. Scientists don't even appreciate this because it is really amazing. I don't feel like I've done this little movie justice, but I have to move on. So again, if you look at the history of life, I always tell people that science is actually a very spiritual. It's kind of good that it's Sunday because science is actually a very, very spiritual exercise. The reason it's very spiritual is because it deals with an completely mysterious fact. We exist. We exist. Yet we don't know where we came from. And we're trying to understand it, right? Science is not about how we got here. We all agree we're here, right? I mean, can we agree on something, please? At least that we are here now. Okay, that's where science starts. If you all agree we're here, how we got here is not as important as how do we work? How does stuff work? What is it made out of? That's what science is about. And it's very humbling because when you start to look, it seems like the more you learn, the less you seem to know, right? And so in a way, it's a really spiritual enterprise, but because it deals with something that's happening right now, it actually brings people together from all over the world. People get together to try to answer questions because we're talking about stuff that's happening right now. How does this work? How does this even work? Science is great because it's trying to answer those very basic questions. Well, here we're guessing. How did life get here? We don't know. But it seems likely, given the nature of the things that are inside of us now, that there was an origin of life in what we call the RNA world, a time when these molecules that could essentially do chemistry, informational molecules that could do chemistry, that could spontaneously add to their chains. They could make copies of themselves, began. It's like this is when the fire started, this inferno that is still burning today. It's thought to have started in this competition, this struggle for survival among molecules. And then that gave rise to organisms that had, for the first time, invented DNA, which it's another form of genetic information. I'll talk about DNA a little bit too. But basically, DNA is kind of like the hardware, and RNA is the software that's running inside of our cells. But then, through the history of life, we have these imagine trillions of lineages in parallel, but talking to each other. This is how we get the diversity of life on the planet, in my opinion. The concept of parent to offspring inheritance is absolutely important for adaptations to local environments that we see. That's how the gazelle gets faster, the giraffe's neck gets longer, etc. But real innovations in biology are often originating in one organism, completely different lineage, and then jumping to other lineages horizontally. And there are great examples. One is the mitochondria. I gave you that. The other is cyanobacteria. They evolved photosynthesis, and then plants and algae obtained chloroplasts, which are remnants now of cyanobacteria that have been endogenized, have been blended into those cells. So rather than having to reinvent photosynthesis, the plants and algae captured cyanobacteria. And by so doing, we're able to capture all the genes necessary to do photosynthesis. And they're doing it now. This is something that we know, biologists know, but I still feel biologists underappreciate that this kind of horizontal transfer of information is truly fundamental to living things. And humans have just started being able to do these types of techniques, right? We've just begun to do gene therapies, fix genes that are broken and put them back, or use mRNA, as you've learned, as a vaccine, and also now these guide RNAs that I'll be telling you more about in a moment. So living things thrive on information that comes from other lineages. And the history of the Earth is so old. I mean, you think a lot of what we see in this more recent part of the Earth's history, this is when all the big stuff was happening. There's almost no fossil record of life below what's called the Cambrian. There's this weird phenomenon called the Cambrian explosion. You've probably heard of it. It was the first time suddenly in the fossil record when there were lots of bigger animals in the sea and also plants. So the Cambrian explosion about 540 million years ago, when that occurred, the organisms that were around before that, we can now infer from their genomes, from the genomic sequences of them, they were already incredibly sophisticated. And when I say that, again, we have a very limited fossil record. And that's because the Earth, we now know, and I'll show in a moment, the Earth was glaciated all the way to the equator below the Cambrian. So there had never been a soil on the planet. There was no land because it was just ice. The living things that were alive were very sophisticated, but they were stuck under ocean ice. And the dinosaur extinction, if you put this on a calendar year, would have been December 27th at 4 a.m. In the present day, January 31st, right? But that should also tell you something, because in just this tiny little bit of time, our own ancestor went from being a little mouse-like creature, rodent, to being, not only us, but think about it, the blue whale, right? It took only 30 million years for mammals to become climax predators in the ocean. And before that, dinosaurs had been the climax predator. There was something called the ichthyosaur that was a huge animal, and they would eat sharks for snacks, right? I mean, the sharks were around the whole time, but for some reason never really, it's the land animals come back into the water and then can become the top predator in the ocean. It's kind of amazing. But mammals did that in a very, very short period of time. Again, organisms are incredibly adaptable. And I would argue that the eukaryotic cell has done something really cool. It's kind of like brought the fire inside. There are these things inside of our DNA, we call them transposons, but they're just like little viruses that are moving around and shuffling the deck constantly inside the eukaryotic cell. And I think of that as sort of bringing that fire inside, because those guys are constantly shuffling the deck, moving stuff around in our genome. Whereas the microbial world, little microbes, they're constantly interacting with each other and exchanging information. They have, in each cell, your average microbe has a tiny genome. But if you count all of the genes that they regularly can access, they actually have a really big genome. They have a distributed genome, meaning their genomes are distributed over, they can even get DNA easily from other species of bacteria. So they are really living in this inferno. This is where those of you who work on this CRISPR phenomenon are probably familiar with this. These are information handling mechanisms that allow bacteria to handle information horizontally, safely, you know. And I like to make that analogy of there's this old lady out there somewhere. She's got 10 million bucks, and she's trying to give it to all of you guys, and you keep deleting her email. But she's really there. There's some really frustrated old lady somewhere in, I don't know, Nairobi or wherever they say there are, and she really just can't give away that money. Living things know that information is valuable, but they also know that it's a double-edged sword. So they're really, really careful about how they handle it. But they can capture information horizontally, and they can silence it. And then they can make another copy of the organism. So imagine your laptop could cell divide, and now you have 2 laptops. Well, then you open that email on one of the 2. You're okay now, right? Well, that's the strategy living things have been taking with information. It is dangerous, and they know it. But without it, without that exchange of information, everyone loses. And herein is why I think the language is universal. It's universal because if you can't speak the language, you are going to lose. You have to be able to. Bacteria speak the human language because the human genome is full of formerly bacterial genes. And bacterial DNA gets vectored between different organisms all the time. Viruses are doing this constantly. Oh, and this is just a picture from NASA, or NSF, I guess, that's showing you what the Earth might have looked like about 640 million years ago below the Cambrian. Little tiny organisms like the worm were under there, and they were already very, very sophisticated. They just had to be tiny. They couldn't be big because there was no land. Yeah, RNA is a software that's running, right? The same hardware, DNA is in every cell, but different cells do different things, in large part because they express different RNA. And what is RNA? Okay, now we're going to get into that. So RNA and DNA both are composed of basically 4 different letters. It's the genetic code. It's a very simple language. You should all know this language because there's only 4 letters in the alphabet. And there's basically just 20 words in the whole dictionary, and I'll go into that in a moment. So now we're shooting inside of a cell, and we're going to go inside the nucleus. This is where the DNA lives. The nuclear DNA, anyway, is inside there. And here you see the DNA is all bundled up very tightly, but now when it's going to be copied, it gets opened up. And now what you're seeing here is what this green blob is a polymerase. There are polymerases that can copy DNA to make DNA, and there are also polymerases that copy the DNA to make RNA. There are also polymerases that copy RNA to make DNA, but that's unimportant for this point. The point here is that you have, in that beautiful staircase double helix, you have these 2 strands that can be used to template each other, right? So this polymerase reads the information in this strand, and it makes a copy. mRNAs are copies of the information that's in the DNA, and then they get processed. So this polymerase will read this information at an incredibly fast, over 50 nucleotides per second, almost flawlessly, copying the genetic information from the DNA. Your cells are doing this right now, making copies of the genetic information. So they slowed it down, 50 per second, just imagine that. And it's incorporating these nucleotides into a chain that will then undergo some processing and be exported out of the nucleus so that it can then be translated. So once it gets processed here, it's going to go out of the nucleus. It's going to encounter something called the ribosome, which are these translating machines that can actually get onto the RNA. This is so oversimplified and it's ridiculous. But the ribosome then can read the genetic code. And the way it does that, it reads 3 nucleotides at a time. Each triplet encodes an amino acid. Oh, and here's the artist's rendition of RNA interference. This is a search protein getting loaded with a guide RNA right there. And then it goes and it searches in the cell and finds targets. And it looks all so stately and slow, but that search mechanism is happening at 100 million interactions per second, the diffusion limit. So our search engine proteins, that I'll show you a little bit more in a moment, are actually really, really fast and sophisticated. Translation, I just was telling you, is the process by which the information in the messenger RNA is read. 3 letters of code spell out an amino acid. So the amino acids are like the words, and they're put into a polymer. So you go from an mRNA polymer to an amino acid polymer. So the ribosome is translating the information. It's taking the mRNA, reading it, and using this universal code that's the same in the bacteria and the human. The bacterial ribosome does the same thing that the human ribosome does. It reads the same 3 letters, puts in the same amino acid. That's why bacteria could make human insulin, because it's using the same language. So the ribosomes are doing this the same way. Now, here, what you're seeing is a protein. And this is a protein that's programmed with a guide RNA in red, and it's a search engine protein. Now, when I was describing this to my family, which you should all, if you're a scientist, you should always talk to your mom. She's full of great ideas, no doubt. Even if she's a scientist, I'm sure she's got great ideas, but if she's an artist, even better. What you're seeing here is the protein. So the spring, the sheets, the strands, these are chains of amino acids that have been then linked together in that process called translation. So as the mRNA was read, amino acids were incorporated into these, into the growing chain. But because amino acids are so chemically diverse, there's 20 different chemistries involved in the different amino acids, you get a much more interesting, much more complex structure. So as RNAs can fold up and they can be complicated and everything, proteins can be way more sophisticated in the way they fold. And what this protein is doing is it's folding up and it's binding very tightly to one end of the RNA. And this red RNA is what we call the guide strand. So it has about 22 nucleotides of information, just 22. But that's more than enough to find targets. And what the protein is doing is it gets programmed, and once it's programmed with a guide strand, this red strand, it then can, what you're seeing here is that you're seeing it bound to a target. So you've got the blue strand here, that's the target. Imagine that it goes off into space on either side. And this is an atomic resolution structure. This is an actual determined structure by Dinshaw Patel's group showing what this complex is thought to look like in vivo. So the protein is binding tightly to one end of a guide RNA. And that guide RNA has about 20 nucleotides of genetic information, which is enough for it to uniquely identify almost any target in the human genome. Think about it. If you want to find the play Macbeth, all you need to remember is to be or not to be. That's it. And you get the whole play. Living things figured that out billions of years ago. There's no living thing on this planet that doesn't have guided search. Everything on the planet has guided search. That's what CRISPR is, if you've been following the news. It's a genome editing mechanism where RNA guides a DNA nuclease to cut DNA. This is now being used to cure sickle cell anemia in humans. Search engines, guided search. It's programmable. Andy and I were amazed when we were injecting these little worms and they started responding to the stuff we were injecting. We knew when we injected them with DNA that we had to put the needle directly into their germline in order for them to take up the DNA. When we injected them with double-stranded RNA, we could miss. Accidentally, my student actually missed the gonad every time. And I told him, oh, nice try, we'll check them tomorrow. Well, he checked them the next day, and I couldn't understand. I came in, he was all excited. Every single one of them worked beautifully. It silenced the gene perfectly, and I'm like, but you missed the gonads. There was a systemic response. The interference was spreading in the animal, and it was being programmed onto these search engines in order to carry out this incredibly ancient mechanism of guided search. So this enzyme, what it does is it shields the guide RNA so that only the first few nucleotides of the guide are presented. Just the first 3 or 4 nucleotides of the guide are presented. If they match, then the protein undergoes a conformational change, exposing more, and so on. And this is the work of many labs. Ian McRae's most recently has done some really beautiful studies on the dynamics of this guided search process. These are the search engine proteins that mediate RNA interference. When they find a target, they actually slice it. Imagine if you could not only search the web, but you could take down the website. That'd be very useful. Living things they know how to deal with information. They are really, really sophisticated, clever. But of course, when we discovered this in the worm, And then we discovered that the genes that work, that do this, that encode these proteins in the worm are also in the human. There was a lot of excitement, because if you could program this type of enzyme to find and regulate a target, what if it's a target that's causing disease? What if this gene is inappropriate expression of this gene is giving you cancer? What if it's giving you high cholesterol or some other, maybe it's making you old or your hair fall out, right? I mean, what if you could turn that gene off and have a healthier outcome for the patient? This was incredibly exciting. So, actually, this is interesting. I forgot this was from 2002. There was a huge amount of hype in 2002 because we had just shown that humans and worms, and we, I mean collectively, the whole field working together, but importantly, the microRNA folks had discovered that there was a human, and a microRNA is a naturally occurring guide RNA, because as you can imagine, this search engine thing isn't just there for us to use. Organisms are using it all the time. In fact, none of you would be alive without it. It's working right now in all of us. Well, wonderful developmental biologists working in the same animal as me discovered a microRNA pathway and showed that these microRNAs, which are tiny naturally encoded guide RNAs, are loaded or are functioning through the same type of protein. And that paper was actually a collaboration between my lab and one of the two people who got the Nobel Prize last year. But in 2002, we published that paper showing that humans have the same, exact same guide RNA mediating pathway that is programmed by this RNA interference pathway. And there was a lot of excitement about it. The news actually picked this up. CBS News wanted to explain how RNAi works. And I did not advise them on this. I'm not taking responsibility. But they came up with this, which looks really good because, you know, you've got the CBS logo and then the beating heart thing. Here's the double-stranded RNA. And now watch carefully. There are the... I mean, how on earth did they get that idea? That is so creative. I mean, RNA cannot chew. But they did get one thing right. I was just telling you, this is an active mechanism. It is like an immune mechanism. The organism is programmed by a double-stranded RNA. If we give you, now there's actual drugs now approved in humans. We can inject a human with double-stranded RNA. It's like the same kind of shot you got with mRNA. But what happens next is truly amazing. The RNA distributes, these guide RNAs distribute throughout your body in your circulation. They get into tissues like the liver, and then they program these search engine proteins that I just showed you that are resident in every cell in your body. Once that protein, that search engine protein is programmed, they silence the target gene for a period of 6 months to a year in humans. So we can dose a human with a single injection or 2 per year and have therapy that continues to knock down a gene that's causing disease. So think about Alzheimer's, think about ALS, think about epilepsy. It works beautifully in the brain. We can deliver siRNAs into the brain. We call them short interfering RNAs. We can deliver them into patients now. It doesn't quite work like that, but it's the same idea, right? It's an active mechanism. Your body makes this mechanism. It is a natural mechanism. Now we can program this natural mechanism with medicines that will, I think, have a dramatic impact on human health going forward because it's relatively cheap. The cost of goods is relatively low. It's not expensive therapy. And I'll tell you just a little bit more about the therapy. I'm not sure how much time I have. 5 minutes? Okay. So with with all biotech things, like with CBS came out with the news article, everybody got really, really excited. This is like called a hype curve. Luckily, the Nobel Committee got very excited. Then all the drugs started to fail. Big pharma sold out. Nothing worked. In humans at first, nothing worked at all. And the reason was that the drugs were brought into the clinic too early. They weren't ready. It took a lot of time. And at first, the drugs that do work now have only been working in the liver. Now there's, I think, 6 approved drugs. The liver is where everything goes first. When you inject it into a patient, you get a lot of drug into the liver. But now there are drugs in development for the brain, as well as multiple other targets. And I'll just tell you a little bit about that. Luckily for us, at the, Andy and I, at the top of the hype curve, the prize was awarded. One of the great things that happened is the state gave us about $100 million to start this RNA Therapeutics Institute. And we recruited Anastasia, a colleague that I'll tell you a little bit more about. We built that building. We started recruiting people who were doing RNA therapeutics, even though there were no such thing. There had never been an RNA therapeutic, but we were the first in the world to have an RNA Therapeutics Institute. So of course we had to make them. So the first thing we had to do is hire chemists, because it takes chemistry to make these molecules function. You have to protect the RNA with certain types of chemical stabilization. But once you do, you have a molecule that is very different. This, like a traditional drug, it's hard to separate the properties of the drug that determine where it goes in the body. They just, because it's a small molecule, it just goes kind of everywhere, and its therapy effect is driven by what it targets and what it binds to. But in this case, the delivery is something that you can work out. You can attach appendages, for example, that determine where it goes. Like the ones that go to the liver, there's like this glycosylated, GalNAc that's added there that helps it go to the liver. But if you want it to go into the brain, you can do other ligands. So you have this concept of a dianophore that's separable from the pharmacophore. The pharmacophore is the sequence. And this is why it's so easy to program these drugs, because all you have to do is change the sequence and you have a new therapy. So this is why the mRNA vaccine is so quickly updated, because you just change the sequence and you get a new therapy. And since I'm kind of running out of time, I'll just mention some of these people very briefly. Neil and Bob are 2 people, incredibly passionate scientists who, medical doctors who seeing patients and just were tired of them dying of Huntington in the case of Neil and ALS in the case of Bob. And they came to UMass in part because they wanted to harness RNA interference for the brain, and have really brought that along. And then Eric and Sonia similarly have been pushing for prion disease therapy. And that's because Sonia herself is a carrier of this rare familial form of prion disease. And when you have this allele, you will die of prion disease eventually. You don't know when, but it's like, you don't have to eat a hamburger to get it. Unfortunately, you're born with it. And when it kicks in, you have a few months to live before your brain is destroyed by this prion mechanism. So Sonia and Eric also collaborated with Anastasia to help break open brain delivery. And they have an open IND right now a new investigational drug trial for their prion therapy. And there's a Huntington therapy and an ALS therapy, all coming from this group of people here. Incredible. Mm-hmm. These 2 you may remember Melissa because she was here in Ohio, I think, a couple of years ago. They met at the bedside where Melissa had preeclampsia. And while she is also my colleague in the RNA Therapeutics Institute, while her doctor was explaining to her what's going on with her placenta, she said, "I think I know how we can fix that." And she came back to UMass, and 17 years later, her son is doing fine. 17 years later, their therapy for preterm babies, preeclampsia, is going into pregnant women this year. So they've made it, and hopefully we're all crossing our fingers. Maybe moms will be able to carry their babies to term. One dose, and that's how safe these drugs are. Can you imagine getting approval for an injectable going into a pregnant woman? That's what we had to do to get this drug. It was hard, and it's now been through the safety trial, is going into an escalating dose trial in pregnant women. So that's incredible. I think I will stop. Preeclampsia is very bad. But I do want to get to the very last slide. You've all seen that. This is my daughter at the Nobel Prize ceremony. She was only 5 years old at the time. And you can see it's a great, if you ever go to the Nobel Prize headquarters, the gold medals are lying everywhere. And I told Vicki, grab as many of those as you can. And it's kind of ironic and poignant because for Vicki to eat the chocolate that's inside those gold medals, she has to give herself insulin because when she was 1.5 years old, she developed type 1 diabetes. She gets insulin made for her by bacteria to treat her condition, and she can live a pretty normal life. It's hard because the insulin is such a difficult therapy. It's dangerous if you give too much. So, and I told her about our siRNA drug, our new RNA interference drugs, and she's like, "Dad, I'll take that. Give me that." Because one injection a year or two per year, as opposed to one after every meal. Whereas in this picture, she's on the pump. She has a pump. She was one of the first kids to get a pump to pump her insulin. Without insulin, she would die. So the people who are on insulin therapy, they are, give me those injections. They are so eager to have this medicine. They are so appreciative of medicine. She's now doing public health research because she wants to try to fix what's wrong with the inequities in healthcare delivery right now. But there's so many new molecular medicines. This is a protein medicine. We now have RNA medicines, guide RNA medicines that are going to change our lives, improve our lives, keep your muscles, bones strong, treat diseases that are caused by single nucleotide changes even. And so there's a really bright future for medicine. And it's so important that audiences like you who are not scientists appreciate it, because these words, RNA, they sound scary. RNA, DNA, what is all this stuff? But living things have been dealing with it for a long time. They're really good at it. And I hope that some of you will have questions, and I would really love it if we had a kid in the audience who's brave enough to ask a question. I'm done talking, but I'd love to talk to you guys more about whatever you have on your mind. Thank you again for having me here.
Okay, so I think Matt's going to bring up the house lights a little bit. I'm going to try to not fall off the stage.
Yeah, please.
There we go. And Dr. Mello will take some questions.
I see one over there. Someone's brave.
And we have some people to run around microphones. Where are we?
Let's start with a kid if there's a kid.
Okay, go ahead, Corinne.
Do you think there's an RNA drug that could help with like Parkinson's?
Yes. They're in development. They're in development right now. multiple different strategies are in development for Parkinson's, Alzheimer's, ALS. The brain, delivery in the brain is so good, every single cell in the brain gets it. Right now, the best approach is delivery into the CSF. It's like a lumbar puncture, and you deliver it that way. But there are new approaches for delivering it from a injection that's more easy. It's like a sub-Q approach. So yeah, there's a lot of exciting research coming on brain therapies. And I hope that we can also treat various types of brain tumors. So far, the cancer treatments have not advanced as far, but mainly because it's harder to do the studies, not because it's impossible, I think. So the future, I think, for brain disorders, including really untreatable types of cancer that right now there's no therapy at all for, maybe we can address with this approach. That's a great question.
Okay, so I think Anita is going to handle it. There we go.
Oh, hi. This is back to the beginning of your presentation. Can you tell me why there's a high percentage of people in the world that cannot get COVID?
That cannot get it?
Not.
Well, I don't know. I mean, maybe they were asymptomatic, but they probably got it. I mean, they were certainly exposed to it. They might have been naturally resistant in some way, but like the kids, a lot of them were infected. They just were totally asymptomatic. Mm-hmm. So these types of viruses are really good at spreading through the air. So I'd be really surprised if they didn't infect people. It's just when you notice the infection, it only happens after the virus is replicating and making, trillions of copies of itself. So exposure to the virus and infection versus being sick are very different things. There are many diseases, viral disorders that are like that, where people just don't even know they're infected, but they're spreading it.
Thanks.
Yes.
Let's see. Go ahead,
Hi.
Dawn.
Hold on. Did I understand you right at the beginning? You were talking about the nucleus, and there's like
Mitochondria.
two nucleuses, one that has the active
Yes.
part and one that has like a storage, like a library of other stuff.
Yeah, the ciliates do that. They have the ciliates, the big single-celled animals that have these amazing cilia to swim around. They have what's called a micronucleus and a macronucleus. The micronucleus has all the DNA. The macronucleus has chopped the DNA into tiny pieces and has only the genes. So as you know, most of our DNA is like transposons and stuff and Did you know that about a third of our DNA is from HIV-related elements called retroelements? These are elements that are RNA viruses that copy their genetic material into DNA, and then they do this really crafty thing. They stick it into your genome. So that's why it's so hard to cure HIV, because the DNA, the virus has incorporated itself into your genome, into your DNA, so you can't get rid of it. That happened over millions and millions of years to all of our DNA. All of us have HIV-like DNA in our genomes. We call them retroelements. So yeah, all that stuff gets chopped out. The ciliates get rid of all that stuff, but they keep it in their micronuclear genome. So their micronucleus is only used once every life cycle when they need to make, they undergo meiosis, they mate. Now they have their micronuclear genomes exchanged. The gametes produced fuse, and then they do all that kind of stuff that we do. But most of their life, or all of their life, the genome that's expressed into proteins is in that macronucleus. So yeah, there's so many different solutions that living things have, but they're really good at handling information and reshuffling the deck constantly.
Let's see. I think, Dawn, there's a fellow here. There we go.
It might be reassuring to know that my 54-year-old niece is also diabetic since age 1.5,
married with 2 children, plays pickleball 4 days a week, is in great health.
That's wonderful to hear. And that's another really amazing thing about insulin therapy, right? I mean, when that drug was developed in the 20s, there were kids literally on their deathbeds who were skeletons at the time because their bodies were wasting away. And some of them took insulin and then they went on to live to be over 80. It's amazing, that insulin therapy is such a lifesaver. And these new drugs are I hope, going to be like that. There are already people who are taking siRNA therapy for genetic disorders that they saw kill their parents. And now they have a shot at a life where their disease will be controlled. It's not a cure. Banting said this very famously. It's not a cure, but it's a therapy that is like a cure because it halts the disease.
I think Anita, go ahead.
Thank you so much for the talk. This is a big picture question, but you talked about your mother pointing to those spheres and saying, study those. There's something important there. Do you have any thoughts on what the next frontiers are for where those next great discoveries are coming from?
Yeah, certainly my mom has a good eye. There's sort of like something beautiful is almost always going to turn out to be important too. But yeah, we don't even know how these things are working yet in terms of how they do surveillance in the germline. But the we call these tiny RNAs that are expressed in the germline, we call them piRNAs. And I don't know why we do that, maybe because that way you guys don't know what we're talking about. But they're weird because there's like 1 million of them in the human germline. And so simultaneously, the cell has so many different search queries going on all at once. And one idea is that they are, because they're intimately involved in regulating the DNA. So the RNA never really gave up control. That RNA world is still in control. Somehow those guided search mechanisms are controlling the way the DNA is packaged. And when you make your eggs, you know how you get a perfect new being, right? This new baby is always, it could be the parents are really old, but the baby looks beautiful every time. Well, how does that happen? Something really special is going on with these guided search mechanisms that are programming the chromatin. How they do that and how that leads to this more robust and pristine genetic state that leads to a flawless baby, that's kind of a frontier right there. I mean, there's a lot of them, but there's so many more of these mechanisms that still remain to be discovered for how organisms handle information. I mean, CRISPR was only discovered a few years ago, 10, 15 years ago. So, okay. Yeah.
So Anita has a questioner, and then there's one right behind you, Corinne, and then I think we'll thank Dr.
I just want to make sure I correctly here. I just want to make sure I correctly understand one thought which I found fascinating. Were you suggesting that the importance, the prevalence of horizontal gene transfer has something to do with sort of the freezing of the central dogma, so that as you would evolve away from it, you get a disadvantage because you're no longer participating in horizontal gene transfer, so it would be anti-adaptive? Was that the thought?
Exactly. Yeah, you got it.
Thank you.
Sure. I could be wrong. I mean, this is it's totally a guess that that's true. I wish that we had another biosphere, but you might predict that if there was another biosphere, the code would also be universal, but it would be different, right? Because it would evolve to be, again, universal, just because being able to share information is so important in the long term. It's also riskier too, though, in a way, right? If you're able to share information from all the other living things that are out there, that means you're also vulnerable to being infected by their viruses, right? I mean, some people think, and I can't prove they're wrong because it's hard to do these experiments, that if you made a life form that had a different code, that it would actually be dangerous. right? Because it could somehow avoid being, anyway, I don't know. It's fun to speculate, but we really just don't know. It is, again, that's the beauty of science. It's dealing with what is, and it's fun to speculate, but at the bottom of the day, at the end of the day, you have to do experiments.
Okay, let's take the last question from over by Green.
Hi, great talk, thank you. So back to the one slide that you had where you had the PG1 or PGL1 tagged with RFP. So the idea of RNAi being in these localized compartments right around the nucleus, is that unique to C. elegans, or is that, because whenever I think of RNA interference, I usually think of microRNAs or siRNAs that are floating around in the cytoplasm. So is that something that kind of evolved first, or is that something that is actually there in all eukaryotes and it just happens?
That's a great question. In the germline, these things, these red balls are very prominent, but not in the somatic tissue. So the germline is special in that it has more of this type of search going on. There's a whole class of small RNAs called the pi-RNAs that in the human, like I said, there's like 1 million different pi-RNAs. C. elegans, there's like 25, 30,000 different pi-RNAs. Whereas the average human cell has only, I don't know, a few different microRNAs at any given moment. MicroRNAs, again, being the guides for the search engine. So somatic cells don't have as much of this going on. And so maybe they have much smaller perinuclear dots, or maybe they don't have any. Maybe it can all happen in the cytoplasm, and certainly Argonauts are just, some of them are quite diffusely located, as you said, out in the cytoplasm. But in the germline, they like to be in the pi-RNAs, or in humans as well, are in the same kinds of perinuclear drops. Yeah.
Is that because epigenetic regulation needs to be really tightly regulated?
Yeah, in fact, what we think, and this is still just a hypothesis, that each of those little drops is different, because underneath each droplet of search engine factors, there's different chromatin. And so there's a membrane, in between, but the RNAs are being exported from heterochromatin into this, and from euchromatin as well, but basically being scanned, and these different little droplets are each different, in fact, in terms of which RNAs are being scanned there. And so they differentiate as well, which is kind of a cool idea, but it's still just an idea.
Okay, Dan.
You guys are great. I love it.
Don't run away yet. Dan, can you come out?
Okay. That was one of the best Q&As I've had in a really long time. You guys are doing a good job of educating your audience here.
So I'm Dan Schoenberg. Craig and I met about a year ago. I was in town. We probably spent an hour and a half on this very topic. But one of the things that came up was I asked Craig to come and give this talk, and he immediately said yes. So please join me in thanking Craig for coming. So this is The Ohio State University, and education is, of course, our goal. And our students and the Center for RNA Biology focus on students is really one of the key things that we do. Students, undergraduates, graduate students, postdocs, they are the next generation. And many of them have come today, several of them, and prepared posters for you. So we invite you to come to the reception. And right now, just around to your left, meet the students, meet Dr. Mello, and have some refreshments. Thank you for coming.
On your way out, I encourage you to pick up one of these flyers.